In chapter 14, we are going to talk about just a couple of items, 14.1 through 14.3. And one of the areas that we are going to talk about is the lac operon. And a little bit of detail of the lac operon will be taken from this chapter 14.5. So selected topics, not the entire topic. So what this chapter is all about is that the bacteria uses many regulatory strategies. And we call this as a cellular regulation. And there are many types, and mainly this regulation can also be associated with many of the signaling processes. Now, here mainly we are going to talk about mainly the transcriptional and the translational regulation, which will regulate the synthesis of different structural genes or proteins, which are different enzymes, and how they will affect the biochemical processes in the cell. So first, let's take a look at these basic definition, constituted and regulated gene expression, and what we mean by these. So the first thing that when we are talking about the genetic elements that is present in an organism, right here in the chromosome, in the DNA, there are some genes which we call as a constitutive genes. And the constitutive genes are the housekeeping genes. And these genes encode the enzyme of the central metabolic pathway. When we're talking about central metabolic pathway, these metabolic pathways are operating all the time. This is the sum of all reactions that is happening all the time in that particular organism. And here we are talking about in the bacterial context. And the cell need constant supply of these active enzymes. And some enzymes, on the other hand, need during the specific conditions on the environments. So the first, when we are talking about the constitutive genes, these number of genes, they are being produced all the time. So if we actually think about, on the other hand, say, animal cell, like an eukaryotic, a human cell, probably in a particular cell, in a tissue, say for an example, about 5 to 8,000 genes are probably operating all the time as constitutive genes and housekeeping genes. And we can think about that some of the genes, like a sodium-potassium pump, or some channels that transport like a sodium or chloride in and out of the cells, these are needed all the time. Or say, for example, glycolytic enzyme, or if it is an aerobic respiratory organism, the genes in the Krebs cycle or electron transport chain, these are operating all the time. So these are the housekeeping and the constitutive genes. On the other hand, there are some enzymes that are only needed during specific conditions and the environment. In an eukaryotic, on the other hand, this could be very specific to a tissue. Say, for an example, a liver enzyme will synthesize some of the enzymes that will never be produced in a nerve cell, or that will never be produced. A nerve cell produce some of these neurotransmitters, which will never be produced in, say, an epithelial cells. So that is different. But here, when we are talking about in the bacterial gene expression, some of the enzymes are needed only during specific conditions, when there is lack of one thing in the environment, or there is abundant supply of one thing in the environment. The genes can regulate the expression of the protein. So why this is important? And the importance is that every process, which is the metabolic process, the transcription, the translation, the production of the proteins, all of them actually require energy. And if a cell doesn't need the excess amount of these proteins or enzymes all the time, if they are produced constantly, it is the wastage of energy. And the cell doesn't allow that. And maintaining a very crucial amount of only what is needed is basically controlled by this cell regulatory mechanism. And usually this is the transcriptional and the translational control, that what we are going to talk about mainly in this chapter. So these genes, instead of the constitutive genes, are called the regulated genes. And we will see that these are the ones which are controlled in these processes. And there are other activities are also in there that we are not talking about. After the translation, the activity of the proteins can still be altered another level, and we call this as a post-translational modification of the proteins, which we are not going to talk about in this process as well. So mainly what happens and how these common regulatory mechanism in bacteria work. The bacteria gets a signal from the environmental stimuli, and there is a sensor which receives and transmits that signal. And in eukaryotics, we also call this as a signal transduction. These amplifies the sensory information, and this is particularly the signal transduction pathway that what we saw. And then there is an effector, and this acts on a genetic switch, which basically alters at the transcriptional level what is being produced in the cell and how much it is produced. And it is just not an on and off switch. This is basically a switch which is just like a regulator, which has a very fine tuning. So this is like not a turn on and turn off bulb switch, but rather you have a dimmable switch, which you can set the intensity of the light bulb in various mode. And the most of the time we are going to see that this is actually achieved in this regulatory mechanism. There is a very fine control about that, not just only on and off, much more than that. So first we are going to talk about the regulation of transcription initiation, and the reason we already talked about that saves considerable energy and the material. And we are going to talk about some of the constitutive, inducible, and repressible genes. And we are going to see that what are some of the DNA motifs that are responsible for this type of regulation. And we're going to see some of the more controlled, like a negative and positive transcriptional regulation in this process. So the first thing that what we are going to talk about, this is in a bacteria like an E. coli. And this was discovered that we are going to talk about this particularly, this system is as a lac operon. But before we go into the detail of that, so here is just an overview of that, what happens with an inducible genes. So this inducible genes is a beta-galactosidase enzymes. Along with that, there will be another two enzymes with that. But due to the environmental nutrient availability, it changes the expression. That means that the beta-galactosidase level can be controlled in the cell like an E. coli. So the function of the beta-galactosidase is that it hydrolyzes the lactose into galactose and glucose. And there is a side reaction of beta-galactosidase, which basically forms, see that the lactose is basically one molecule of glucose associated with a galactose with a beta-1,4 linkage. This is a beta-1,4 linkage. Whereas this side reaction of beta-galactosidase sometimes produce a beta-1,6 linkage. So if you just calculate the carbon, it is 1, 2, 3, 4, 5, and 6. It is a 1, 2, 3, 4, 5, and 6. So this is your 1 and 4. Here it is 1, 2, 3, 4, 5, and 6. So this is beta-1,6 linkage. This molecule is known as allolactose. But the end product is basically the galactose and the glucose that can be utilized by the cell for the energy production. Now, in the absence of lactose in the environment, you have only about approximately 3 beta-galactosidase molecule per cell in an E. coli. But the number of beta-galactosidase molecule can increase to 3,000 approximately in presence of lactose. And this is basically called the inducible gene expression. And we are going to talk in detail about that. So there are some terms that is associated with these type of regulation, and we call this as an induction and a repression of enzyme synthesis. Induction means that when necessary, we are turning on a gene. That is induction. On the other hand, if there is a gene that is already turned on, repression means that we are turning off that gene. So on the other hand, an inducer molecule is an effector molecule that stimulates the gene expression or turns the gene on. Inducible genes that encode an inducible enzymes, that means that can be turned on from off. And this is required only when the substrate is available. The reason is that if the substrate is not available, if you are turning on this gene, then it is a wastage of energy. and that is not done. So only when the substrate is available, the enzyme is produced, and this is inducible genes. On the other hand, there is another gene which is known as a repressible genes. These genes are involved in many biosynthetic pathway, and they are usually turned on. But if the product of that biosynthetic pathway is enough, then what happens, that product basically repress or turns off that gene until it is again necessary. So on the other hand, there is a word which is called a repressor or a co-repressor. These are small effector molecule which can bind to certain elements and can repress or turn off gene expression. So again, two things: inducible genes are usually off, can be turned on. Repressible genes are usually on and can be turned off if there are too much of the product produced, and excess of that product will be basically wastage. So once it is turned off, that repressible genes can be turned back on again. And this is basically the heart of regulation. So there are many regulatory proteins that basically control the transcriptional initiation. And we are going to see that this control basically happens when the sigma subunit of the RNA polymerase position the core enzyme at a promoter. And there are many transcription factors which are DNA binding regulatory protein. And they basically bind into a specific area where you find an helix turn helix. And usually most of these transcription factors do have some characteristics like this, which does have also a structure, dimeric structure, which basically places them or kind of clamps them onto the DNA, particularly the major groove of the DNA. So another part of the protein interacts with the effector molecule, and one interacts with the DNA. So you have two parts of it, and we will basically explain that what we mean by that one is a DNA binding domain and another is an effector binding domain about that. So this is a classic example of a typical transcription factor with a general helix turn helix motif. So here you are looking at the double-stranded DNA. You can see that these are the major grooves of the DNA. These are the minor grooves. And now you can see that there are two proteins. One is on this side and another is one side, which are dimeric. And there are two recognition helix which form each subunit and interacts with the major groove of the DNA. What do we mean by that? See right here, this is an alpha helix. Then you have a turn and you have an alpha helix. So this is interact with the sequences here. This is interacting with the sequences here. Then you have an helix turn helix interacting with this sequence and this sequence. And each of the helix, that 1, 2, and 3, and 4, recognize what we call as a palindromic sequence. And the palindromes is basically that you read forward and backward the same way. So suppose this is a 5' to 3' of your DNA molecule, and it reads by like this. It is AACAC. So you have AACAC that is recognized by this. Then you have a gap. Then you have another sequence which is basically on the other strand, which is AACAC. So that is recognized by the other side. So that's basically one thing. On the other hand, here you can see that this is also binding to this region, which will basically see this way, that one of them will basically recognize this TTGTG in the 3' to 5' strand. And on the backwards, on this strand, you have a TTGTG that will be recognized by the other helix. So that's basically that how the two recognition helices in general recognize the DNA binding domains. And this is a typical structure of a transcription factor. So now we are looking at the overview of the transcriptional control, negative control and positive control. Here what we are looking at, that this is the part of the DNA, and there is a region which is the promoter. The promoter is the region where the RNA polymerase actually comes in and binds. Close to that region, there is another region in the DNA which is known as the operator. And the operator, on the other hand, binds to a transcription factor which is known as a repressor. If a repressor protein binds onto the operator, it basically blocks the RNA polymerase to initiate transcription. So this is a repressor protein, and this is called a negative control. So if you want to keep a gene off, then you need a repressor molecule binding to the operator, and RNA polymerase is not going to work. On the other hand, what is the positive control? Positive control, on the other hand, binds to another site, which is an activator binding site. And the activator binding to that site actually enhances the binding of the RNA polymerase onto the promoter. And usually this is upstream of the promoter, encourages the RNA polymerase to bind. And this is a positive control. And there are many things in between that we are going to see. This is the very basics of it. So on the other hand, the gene expression rarely is all or nothing that we were talking about, that it is usually like a dimmable switch. And we are going to introduce a term, we did not introduce the term yet, that operons, which are basically a collection of genes that is related to each other for a particular metabolic function. They are related. And these basically make an operon, and they are next to each other, these genes. And the operon sometimes considered as leaky, meaning there is always a low basal level of transcription. And what it does, that low leaky transcription basically can make these small amount of beta galactosidase. So see that there is not that there is completely absence of beta galactosidase, but when it turns on, it produces a lot more. So the production of this small amount of beta galactosidase is a result of the leaky basal level of expression. So an operon is basically a structural genes, which structural genes means that do not always think that they are going to make a specific structure in the cell or something. Structural genes means that this could function as an enzyme together, and together with an upstream regulatory region is called an operon. And the promoter, which which is the binding site for the RNA polymerase, can have different strengths. That you can say that it's a strong promoter, or like a not so strong promoter. That means that depending upon that how tightly a promoter binds to the RNA polymerase, the amount of gene expression can also vary, affect the frequency of initiation of transcription. So here, first, we are going to discuss the negative control of the lac operon. The lactose operon, but first discovered by the Francis Jacob and Monod, Jack Monod. And the Jacob and Monod, basically, they received Nobel Prize in 1965 for the discovery of the lac operon. So you can just well imagine that this was the first elucidation of regulation of gene expression, how a gene expression or production of the end product of a gene, how it can be controlled. So the lac operon contains 3 structural genes. The first one is known as LAC-Y, that encodes a lactose permease that allows the movement of the lactose from the environment through the membrane into the cell. So this is your lactose permease. So which is basically the transporter. So you need a transporter to transport that is your lactose permease. So this is basically the structure of the lac operon, and it contains 3 structural genes. So the first one is known as a LAC-Z. This encodes beta galactosidase. This breaks down the lactose into glucose and galactose, and as a side reaction also produce allolactose with a beta-1,6 linkage in between the galactose and glucose. The second enzyme is a LAC-Y. This encodes the lactose permease, and we have seen this picture. This is responsible for bringing lactose from the environment through the plasma membrane. This is a transporter into the cell. So this is your lactose permease. And then there is the next enzyme, which is known as a LAC-A, which is known as beta galactosidase transacetylase, which acetylase, the product, which is a galactoside product, means that it from the acetyl-CoA enzyme, it puts these acetyl group into the glucose and galactose, and the function for that, why they're doing this, is not very clearly understood. But we clearly understand the function of these two. But these are all related genes. They are next to each other. And upstream, which is basically before the start of the lag Z, there is an operator site, there is a promoter site, which basically binds the RNA polymerase. And then we're going to see that this is a activator site we know as a CAP, which is a catabolic activator protein binding site, CAP site. This is the activator site. We talked about this activator site right here, and it's right there. And before that, you have a regulatory gene known as LAC-I, which produces a lactose repressor. This is a transcription factor for this. So we are going to see that. And this is controlled by a promoter, which is a LAC-I. So this LAC-I promoter only controls this, whereas the LAC promoter, CAP site, and operator controls the expression of all three genes. So the enzymes, the LAC-Z, Y, and A, is not produced all the time. On the other hand, this LAC-I promoter is always active, and the LAC-I, which produce lactose repressor, pretty much all the time. So the enzyme is not normally produced, which are these beta-galactosidase, lactose permease, and transacetylase. They are not produced unless lactose is present. So on the other hand, the LAC-I binds to the operator region, the repressor, inhibits the transcription when there is no lactose. So this lactose repressor binds to the operator and inhibits transcription. And how this happens? So in the absence of lactose, that each dimer of the tetramer bind to the 3 operator site, which is operator 1, 2, and 3 region. So you can see that right here, you have an operator 1. This is your LAC-Z, and this is an operator 2 and an operator 3. So right here, you are looking at a LAC repressor, which is basically a tetramer. So what happens? These are the interacting domains, and you can see that in here, the LAC repressor is binding to the operator 3 region right here and the operator 1 region. And it is completely occluding the promoter by making this loop. So even if the RNA polymerase try to bind in here, it cannot go anywhere. So basically, the transcription is blocked. On the other hand, it can also form a loop in between O1 and O2, O1 and O2, and the DNA basically bends right here. So even if the RNA polymerase tries to come over here, it has to pause because it cannot go through these LAC repressor produced loop basically. So here is a molecular rendering of that. The red one is the DNA. And here you can see the structure of the LAC repressor. And you can see that right here is the bending of the DNA. So you can see that this is a helix turn helix loop, helix turn helix loop, which basically recognizes the palindromic sequences present in these operator areas. So basically what happens that in the absence of the lactose, they basically slide along the DNA, and they find at the operator site, and then they make these kind of loops. So when they are bound, the genes which are LAC-Z and the other related genes, or the LAC-Z, Y, and A, they are not produced. So here, first we are looking at that when lactose is absent, these LAC repressor is constantly produced because LAC-I is always an active gene. It's producing mRNA translated into the LAC repressor, which is active, bind to this operator region, and the promoter region is occluded. And it cannot basically allow the RNA polymerase to transcribe. So that's basically the repression. That means that there is no transcription of LAC-Z, Y, and A. And also always remember that no lactose available. Even in that case, what happens, there is a very low basal level of transcription that can happen. And why this is? Because remember that these molecules are being produced and they are bumping around and finding this. Sometimes there could be some instances where this can fall off temporarily from these bindings. And at that time, the RNA polymerase can move and produce the mRNA for these, and that can be translated into the protein. And that is very little. And as I said, that when this basal level is present, it just produces about, say, 3-4 molecules of LAC-Z. So that is your leaky expression, or very, very low basal leaky expression. And we almost can think about that they are basically not produced in the absence of lactose, except the leaky expression, just a few molecules. Now what we are seeing that when the lactose repressor regulation, when lactose is present. So as we said, that when there is no lactose, even in that condition, there is a slight production of each one of them, which is the lactose permease and beta galactosidase. So what happens, these lactose permeases are present on the plasma membrane. And if lactose is present, even 1 or 2 lactose permease will bring lactose into the cell. And even that small amount of lactose that will get into the cell, that small number, like 3-4 molecules of beta galactosidase will find them, and it will break down them to lactose, glucose, lactose to glucose and galactose. And also it will convert some of the lactose to allolactose as a side reaction that we have seen with a beta-1,6 linkage. So these few molecules of allolactose now will bind to the repressor molecule. So it will bind to the repressor molecule. And when the repressor molecule, which is the allolactose is the repressor, when that is binding to this allolactose, the allolactose is basically, you can say that this is a co-repressor. When this allolactose will bind to this repressor molecule, which is produced by the lac-I, this is basically inactive. So right here is a repressor molecule which can bind to the operator region O1, O2, O3. Allolactose co-repressor binds to this, and see that these are these double structures. That means these are basically galactose and glucose bonded with a beta-1,6 linkage. It renders the repressor molecule inactive. So now no longer the repressor can bind to the operator region. So at that time, the operator is free, and the RNA polymerase will be recruited into the promoter region, and it will move on to move the transcription. So it will increase the LAC-Z, Y, and A production. So as more lactose permease is produced, more lactose permease will go into the plasma membrane to bring more and more lactose into the cell. Now you have more beta-galactosidase. More beta-galactosidase is going to break down them to glucose galactose, and more allolactose is going to be produced. More allolactose is going to bind to all the repressor molecule, and gradually there will be enough amount of allolactose to repress all the produced repressor molecule, and the transcription will pick up, which is present of the allolactose. So they will be produced in large amount to utilize the lactose in the environment. So this is the lac repressor regulation. Now, we are going to see that there is also another type of regulation that can happen, which is with the tryptophan operon. And with the tryptophan operon, there are 5 structural genes. So if we just look at the TRP operon, you have this TRP E, TRP D, TRP C, TRP B, and the TRP A. And they produce different things like anthranilate synthetase, produce anthranilate, and cooperates, the TRP D cooperates with TRP E. And this is the phosphoribosyl anthranilate isomerase. It does a 2-step reaction. And then you have a first subunit A of the subunit of the tryptophan synthetase, and this is another subunit of tryptophan synthetase. And basically, what happens that they all combine the product of serine to produce the serine amino acid to produce the tryptophan. But that detail is not necessary. But just remember that this TRP E, D, C, B, and A. These are all related proteins in the tryptophan biosynthesis, which is a crucial amino acid. Not very common, but still it is required. So now what you can see that this is the operator region O. This is the P, the green one is the promoter region. And right here is the TRPR, which is producing the TRPR mRNA and produces an inactive TRP repressor. So this tryptophan operon, on the other hand, is opposite to the lac operon. So if you just remember that, see, the lac operon is always turned off in the absence of lactose, and it is an inducible operon. That means it is turned on in when lactose is present. So this is an inducible operon. Whereas the tryptophan operon is the opposite, which is on all the time, and it can be turned off if too much tryptophan is produced. So this is a repressible operon. So we talked about the structural genes, negative transcriptional control by the TRP repressor. Operon only functions in absence of tryptophan, and when the TRP is present, it acts as a co-repressor. So what is happening that in a normal condition, when the tryptophan is being produced, all the transcription is going on, and this is basically the inactive TRP receptor. There is nothing. Now what happens if there is a high tryptophan level? Then only the tryptophan repression, the TRP operon repression occurs. So right here you are looking at the tryptophan molecule. It binds to the repressor. So you can see that right here, the tryptophan by itself, which is the product of these operon, is by itself a repressor molecule, which is called a co-repressor. So this is a co-repressor, which is tryptophan, and this is the repressor molecule. And the co-repressor and repressor forms an active complex. And the co-repressor binds to the operator region, and the transcription of these TRP operon is turned off. So this is basically that with the low tryptophan level, transcription of the entire TRP operon occurs. This is constitutively on all the time. When there is too much tryptophan, it can be turned off. So this is basically a repressible operon, just the opposite of an inducible operon of the lac operon. Now what we are going to see that we just talked about just a very basic control of two things, lactose absent and lactose present, two conditions. Nothing is happening when it is absent, and lactose present, there is a production of these. Now there is a little bit of finer control, and that we actually talked about. And this basically depends upon the presence of glucose in the environment or not, and how this is basically controlled. And this portion is from the chapter 14.4, and you will be able to see that in that area you will see the catabolic repression, and this is for next few slides are from that area. So there is a molecule called the cyclic AMP that can further bind to a catabolic activator protein and fine control the production of the lac operon products. And this relies on the phosphoenol pyruvate sugar phosphotransferase system, which is known as a PTS. And the phosphorylation state of the PTS is an indicator of glucose abundance. So what happens that when there is a low glucose, this cyclic AMP is produced, and we are going to see that how it happens. And the enzyme system transfer the phosphate to adenyl cyclase, and the CAMP is made. On the other hand, when there is an available glucose, the glucose that comes into the cell is converted to glucose 6-phosphate, and the enzyme system transfer the phosphate to glucose, which forms the glucose 6-phosphate, and CAMP is not produced. So what happens? Production of CAMP or absence of CAMP actually control the lac operon in some way as well, and we are going to see that. So first, let's take a look at how the glucose, presence of and absence of glucose, how it is basically understood by the cell. So here you are looking at a system right here where you are looking at these 2B and the 2C. These are the transporter of the glucose on the plasma membrane, and enzyme 2B, 2C complex. And right here, this is a phosphotransferase system where phosphoenol pyruvate, which does have a phosphate, is converted to pyruvate and transfers the phosphate to an enzyme system known as a E1. So now from E1, it is phosphorylated. Now this the E1 enzyme phosphate complex now transfers this one to an histidine phosphocarrier protein. So this is an histidine phosphocarrier protein that is phosphorylated. And the histidine phosphocarrier protein actually transfers it to an E2A system, which is now phosphorylated, and HBr phosphorylated, it becomes HBr. So this is just very self-explanatory. So now you have this charged E2A system with a phosphotransferase. with a phosphate, not phosphotransferase. This whole system is phosphotransferase system. Now, this molecule, when glucose is abundant in the environment and glucose is coming into the cell, this phosphate is transferred to the glucose to form glucose 6-phosphate. So, which in turn transfer it to the incoming glucose. But if glucose is absent, this E2, phosphorylated E2, there is no place for this phosphate to go. Instead, what it does, that there is another enzyme which is associated with the membrane. This is known as adenylate cyclase. This E2A phosphorylates the adenylate cyclase by transferring the phosphate to this, and this is activated. And what happens, that activation of the adenylate cyclase basically produces the cyclic AMP. So ATP is broken down to the cyclic AMP. And cyclic AMP is known as a second messenger molecule, which is basically a signaling molecule into the cell, and it will bind to another protein and it will tell it or destine it to a specific function or a control regulation. So when glucose is abundant, there is no cyclic AMP production. When glucose is not there, this is how the cyclic AMP is produced. And what is basically cyclic AMP? So you can see that right here is the ATP. You have right here is the ribose sugar. This is the first carbon, and right here is the adenine, and you have 1, 2, 3, 4, and 5. It's a pentose sugar, and right here is the fifth carbon, and you have your alpha, beta, and the gamma phosphate. This is your ATP. Adenyl cyclase removes these two phosphates, and you basically have an adenosine monophosphate, but this is just not a regular adenosine monophosphate with just a phosphate. This basically make this molecule as a cyclic molecule where this phosphate, 5' phosphate, is attached to the 3' hydroxyl, and this becomes a cyclic molecule. This is the reason we call this as a cyclic AMP, adenosine monophosphate. This is a powerful signaling molecule. So now what happens, this cyclic AMP binds to a protein which is known as a CAP, or catabolic activator protein. And this regulates the lac operon in presence or absence of glucose. And we are going to see that this catabolic repression allows the E. coli to use glucose preferentially over all other carbon energy sources. So we are going to see that how the E. coli basically fine-tune the use of glucose over all other, like lactose, in all cases. So let's take a look at this. So now we are looking at that how we already learned that what the lac repressor and the allolactose is doing. But now we will concentrate on the CAP site, which is a catabolic activator protein binding site or activator site as well. So there are 4 scenarios that can happen in here, and I'm going to describe it through the picture. So here in this case, there is lactose present, but there is no glucose in the environment. So as an energy source, the E. coli has to depend only on lactose and break it down to galactose and glucose and get the glucose from for the glycolytic pathway. So now what we are seeing, that in that case, as there is no glucose, remember that in absence of glucose, the cyclic AMP is produced, and the cyclic AMP binds to this catabolic activator protein, which binds to the CAP site. On the other hand, lactose is present as well. So allolactose is produced, and allolactose binds to the lac repressor. So lac repressor cannot bind to the operator region. So what happens in here, as the activator site is occupied by the CAP-CAMP complex, this allows very, very tight binding of the RNA polymerase to the promoter. Basically, it recruiting efficiency increases, and there will be a lot of transcription. This is the highest level of transcription, which is basically the production of the lac operon genes ZYA will be the highest in this level, where only lactose is present, but there is no glucose. Now, what happens? The next case is lactose and glucose both are present in this case. And as we said, that the catabolic repression allows E. coli to use glucose preferentially over all carbon energy sources. That means lactose. So when glucose and lactose both are present, E. coli still wants to use glucose first before it can go to lactose. So what's going to happen here? You can see that as glucose is present, there will be no cyclic AMP production because the glucose is going to convert it to glucose 6-phosphate by the E2A enzyme, and adenyl cyclase will not be phosphorylated to produce the CAMP. So no cyclic AMP, and the cap protein is inactive and it cannot bind to the activator site. On the other hand, there is as the lactose is present, a small amount of lactose is going to come into the cell, convert it into the allolactose, and the repressor is going to bind those. So here the repressor, some of the repressor are not going to bind to the operator region. On the other hand, the cap is inactive, so some of the transcription will go on, but this is a low level of transcription, just a low level of transcription, not as high, because here the activator protein recruits the RNA polymerase tightly. Here it is just a little bit low, but still a little bit of transcription of the lactose operon can go on. Now here is a condition where there is no lactose, no glucose. So if there is no glucose, the cyclic AMP is produced and that binds to the cap protein, and activator site is occupied by the cap protein. But there is almost no lactose, so the allolactose is not produced in the cell, and the lac repressor cannot be inhibited by the allolactose. So the lac repressor, which is produced by the lac-I, now binds to the promoter, and the RNA polymerase, even if the cap site is occupied, even if in that condition, the RNA polymerase recruited on the promoter cannot move on because of the DNA bending by the lac repressor. So here, the level is almost absent, or we can say that only maybe the leaky expression, just a few molecules, 1, 2, 3 molecules, if the lac repressor by itself falls off. So very, very low, the lowest amount in of leaky expression can happen, almost absent, we can say that. On the other hand, the fourth case could be that where glucose is present, but there is no lactose. So in that case, what happens? You can easily see that there is no cyclic AMP production. So in that case, the cap remains inactive. On the other hand, what happens that as lactose is not present, it cannot bring in the lactose to form allolactose, and the lac-I repressor cannot be inhibited by the presence of allolactose, and it binds to the operator, and the lactose operon is basically turned off in this way as well. So in this case also the cap is inactive. There is almost absence of any transcription of the lac operon. So the reason for that, because you have plenty of glucose. So this condition, you have neither glucose, there is nothing in here. So you have to use a different carbon energy source. Here you have glucose, so E. coli is using the glucose. There is no need for the lactose as it's not in the environment. And that's the reason that the transcription of the lac operon is absent in here, or maybe just a basal level of transcription that can go on. So this is how there are different levels of transcription. And you can see that it is more like a dimmer switch of control rather than just turning on and off. So there is much more finer control of the repression with the help of the catabolic activator protein in presence of glucose or lactose. The next, we are going to talk about this arabinose operon. This is also known as an ARA or ARA-bad operon. So arabinose is a sugar which is essential for to make a product, xylulose 5-phosphate, which is essential for the pentose phosphate pathway and nucleotide production as well. So regulatory ARA-C is the gene which is controlled by this para-C. This is the promoter. And on each side of the promoter, you have a control site, which is an operator site, ARA-O1 operator site, ARA-O2 operator site. Then there is a catabolic activator binding domain. As we have seen, this is the same thing, which binds to the CAMP and catabolic activator protein. followed by an era I1 and I2 region. And this is just shown as an era I, just as a single region in your textbook. But that really doesn't matter. It's the same thing. So these are the control regions. And then you have the promoter, which is the para-bad promoters, and the recruitment site for the RNA polymerase. And then you have the structural genes for the arabinose operon. So the first one is ARA B, that produces this ribonucleokinase. ARA A produces an isomerase, and then ARA D, which is a kind of an isomerase, which is known as an epimerase, basically. So essentially what happens, this converts, these enzymes converts, as I said, arabinose to a xylulose 5-phosphate, which takes part in the pentose phosphate pathway. So the para-bad chromatal 3 structural genes, then ARA C protein that is produced from this promoter acts as a repressor in absence of arabinose, and ARA C protein also acts as the activator in presence of arabinose. So very interesting. And ARA C also autoregulates its expression when there is a high ARA C level. So one protein can do many things. It can be a repressor, activator, and an autoregulator, or autorepressor or autoregulator. So many functions can be done with this. So let's take a look at that, how they are associated with the DNA. So here we are looking at the structure of the DNA, and this case, this A operon is inhibited in the absence of arabinose. And let's take a look at what is basically happening. So this large molecule that you are seeing, this is basically the ARA C, which is produced from this ARA C gene under the control of this ARA C promoter. So this is your ARA O1 site, this is your ARA O2 site, this is your ARA I site, which is divided into ARA I1 and 2, so ARA I site. So this ARA C protein, this large protein, one molecule binds to the ARA O2 and another molecule binds to the ARA I, and they dimerize. So you can see that this is a linker region, and they dimerize. And as they dimerize, they bend the DNA like this. And see that there are also 2 molecules of arabinose are binding to this ARA O1 site. So due to this bending of the DNA, basically the RNA polymerase cannot be recruited onto the promoter, and that's why the transcription of the ARA B, A, D, this cannot. So this RNA polymerase cannot bind in there due to this bending of the DNA. So in this case, when the operon inhibited in the absence of arabinose, that means when arabinose is not present in the environment, the transcription is absent. RNA pol can't access the para-bad promoter due to this bending. Now we have a case where the arabinose is present. When arabinose is present, this pink molecule is arabinose. And see that there is an arabinose binding site in the ARA C protein, these empty spaces right here. So that is occupied by the arabinose when there is arabinose binding domain right here. So when arabinose bind to these molecules, very interestingly, the interaction between ARA C protein bound to ARA C and the ARA O2 bound to ARA C, this linkage is broken. Because what happens this linker domain right here, due to the binding of arabinose, this actually bends. And that's the reason that this will basically separate these 2 molecules. So you can see that right here, arabinose binding separate these 2 molecules, and the loop is basically broken. And on the other hand, what can happen in that condition, that the ARA I, and as we said, that why there are 2 molecules, that will explain that, because you have an ARA I1 and ARA I2, 2 regions. So one ARA C can bind here and another ARA C can bind here. And in the ARA O1 region, also 2 molecules of ARA C can bind, which you are seeing in there. And they can interact with each other as well. But these basically doesn't inhibit the recruitment of the RNA polymerase. The loop is broken. And this, in fact, promotes the recruitment of the RNA polymerase, and it goes on transcribing era B, era D, and era D. So here what you are seeing, that in the absence of arabinose, the era C is working as a repressor. On the other hand, in the presence of arabinose, the era C protein acts as an activator, which helps the recruitment. So these basically helps the recruitment. The operator, when is dimerized, the dimerized era C with the arabinose is present, it helps the recruitment of the RNA polymerase into the PBAD promoter and helps transcription. So that is another thing. So transcription present, RNA pol can access PRBAD promoter. Now what happens that when there is absence of glucose, you produce cyclic AMP. And when the cyclic AMP is produced in absence of glucose, it forms the binds to the catabolic activator protein, which is next to the era I region. So even the recruitment of the catabolic activator protein even can enhance the binding of the RNA polymerase. So it can even increase the amount of further enhance the transcription of the arabinose in absence of glucose. So in this first case, arabinose is absent. Second, when the only arabinose is present, this cap binding doesn't happen. There is a transcription of the era B, era A, and era D that happens. When there is absence of glucose due to the cap binding, the transcription level even further increases. So this is basically the arabinose operon, working of the arabinose operon. So after in the 14.3, we are going to look at the attenuation and the ribose switches, which can also stop transcription prematurely. So we already talked about the trip operon producing the tryptophan, and we already talked about the regulation of that. And here we are going to see that even that trip operon can be controlled after the transcription has been initiated. And this is very, very interesting process, and why this is important. So what happens that we are going to see that the transcription can also be regulated by controlling the transcription termination. And the way this is controlled, this mRNA does have a very long leader region, and this leader region is before the first structural gene of the trip operon. What do we mean by that? We will take a look at this in a second. And the RNA folding patterns in the leader affects the transcription termination. And this type of regulation is known as the attenuation of transcription, and was first demonstrated by Lee and Janovsky in the trip operon. And then we are going to look at the ribose switches. So why this is important, but just think about this way, that just the trip operon that we just talked about right here, this can reduce right here the amount of tryptophan produced by almost 60 to 70 fold. On the other hand, on top of that 60 to 70 fold, the transcription attenuation can even further reduce it down to 10-fold. That means overall, you are achieving about 600 or 700-fold reduction in transcription. So this is a huge advantage of that to turn off the transcription. So let's take a look at how the trip operon transcription actually happens. So if we are looking at the trip operon, it's a little bit more detail here. So here you have the trip R, which produces right here, the repressor, and this is the promoter for the trip R. Then right here is the promoter for the trip operon. Then you have the operator. This is the leader sequence, which does have a start codon and a stop codon and can produce a leader peptide. And after that, you will have the trip operon right here. You will have a start site for the trip A, stop site, start sites, and those. And these are all polycistronic mRNA. You know that the polycistronic mRNAs are produced in bacteria. We have already seen that. And what these are the enzymes that are produced. You produce TRPE, produce anthranilate synthetase. This produce anthranilate phosphoribosyl transferase. And there are two enzymes that are produced by the TRPC, phosphoribosyl anthranilate isomerase and indole triglycerol phosphate synthetase. Then you have the two molecules of tryptophan synthetase B protein and A protein, two different molecules of that. So this is basically the entire trip operon. So what we are looking at, that the attenuation is basically a termination of transcription within the leader region or where the leader peptide is produced. And this occurs through a stem loop structure in the leader RNA, depending upon the tryptophan level in the cell. And one of the most important thing you have to remember in here, remember that this type of attenuation can only happen in the bacteria because transcription and translation is coupled in bacteria. What do we mean by this? So the mRNA is being produced in the cytosol, and the ribosome that binds to that mRNA, as it is being transcribed from the DNA almost immediately, RNA polymerase goes on and making the mRNA, RNA polymerase. Behind that, the ribosome is translating into the protein. So translation and transcription can happen at the same time. So we call this as a co-translational transcription. This does not happen in eukaryotes. The reason for that, in the eukaryote, you have the nucleus, and the mRNA is produced in the nucleus, and then they are processed by many different ways. They are spliced to remove the introns, then they are polyadenylated, they are capped, and all these things happen to the mRNA. Then they are transported through the nuclear pore into the cytoplasm before they can be translated into the proteins. So translation and transcription is not coupled in eukaryotes. So this type of transcriptional regulation doesn't happen, or attenuation doesn't happen in eukaryotes. This is particularly for the bacterial system where the translation and transcription is coupled. So let's take a look at how it works. And to understand this, we have to see that the different regions. So here we are looking at only the TRPE region. Remember that. After that, you have D, C, B, and A. We were just seeing that. And basically, this region is the leader region. So in the leader region of the DNA, what you have, that you have regions, this 1, 2, 3, and 4. This region 1, 2 can form stem loops. 2, 3 can form stem loops, and 3, 4 can stem loops because they have complementarity. So you have possible loops 1, 2, 2, 3, 3, 4. And in this region, there are 2 trip codons, or the tryptophan codons, next to each other. One tryptophan codon, another tryptophan codon. Very unusual. Usually, tryptophan is a very rare amino acid, and it occurs just only on an average over 1 every 100 codons. Here, they are just next to each other. And right here, this you call as in between 1 and 2, the pause loop. There is an UGA stop codon right here. This is the end of the leader. So a leader peptide is produced, and the leader peptide will stop right here. And this is just before the UGA stop codon is just before the region 2. Region 2. So UGA stop codon just before region 2. And pause loop right here. And this in between the loop, in between 3 and 4, this is known as a terminator loop. And just remember that this is also very rich in GC. So these are tight loops. And this region 4 is followed by a poly-U tract. So you have U like this. So basically that's in the RNA. So basically when it transcribes from the DNA to the RNA, just remember that there are lots of A's in here, and that's why they are putting the complementary U, and this is a poly-U tract. And this is a universal thing from all the RNA polymerases. The RNA polymerases tend to slip over a poly-U tract. And there are many examples from viruses to eukaryotes. This happens all the time. If you have too many U residues one after another, that's called a slippery sequence where RNA polymerase actually slips. So that's in there. So this is basically the features of the RNA that is being transcribed from the DNA by the RNA polymerase. So RNA polymerase is basically moving along, and they are making this leader RNA. This leader RNA is very unusual, very long, about 162 base pair. This is before the first codon of the operon region, the first gene of the operon, basically, which is TRPE, so pretty long. And this long leader sequence, basically with the secondary structure, cause this attenuation. So what are the different things that can happen? So in this condition, if you have a low tryptophan level, let's see that how attenuation can happen in the low tryptophan level. So tryptophan level low means that the RNA polymerase need to transcribe all the trip operon genes to produce more tryptophan. So in this case, what is happening? Remember that the RNA polymerase starts transcribing, and as the transcription and translation is coordinated, or they are co-translational translation, the ribosome follows the RNA polymerase and it translates the protein. RNA polymerase is reading the DNA, making the mRNA, and the ribosome is translating it into the protein concurrently. It is just a little bit behind. So now what happens? That RNA polymerase is going on, and right here, the ribosome now comes into this region where it finds in this region this tryptophan codon. So you can see that there are three sites. You have the exit site, then you have the peptidyl site, and then you have the acceptor or amino acid site. And this is the first tryptophan codon. It comes in here. Now, followed by that, you have another tryptophan codon just after that. But remember that when you have a low tryptophan level, you do not have much charged tryptophan tRNAs in the cell, due to lack of charged tryptophan tRNA. So as the tryptophan tRNAs cannot occupy this space immediately because there are two trip codons one after another, the ribosome actually stalls in this region 1. So as the ribosome stalls in the region 1, on the other hand, the RNA polymerase goes through the region 2, goes through the region 3. As soon as RNA polymerase travels over the region 3, the 2,3 can form stem loops. So 2,3 immediately can form stem loop structure. As 2,3 already formed the stem loop structure, the region 4 remains like this, and the RNA polymerase goes on and transcribe the entire other genes of the trip operon. And then gradually what will happen? The ribosome will catch up. It will read through this trip operon, and it will just make the leader peptide, and then the entire mRNA is produced. So it will just go over and basically translate all the genes required for that. So in this case, there is no attenuation of transcription. RNA pol continues to transcribe the TRP structural genes, which will be basically produced into the protein, and those proteins will synthesize the tryptophan to bring it back to the higher level. So this is no transcriptional attenuation. So now the next in what is happening, that when there is a high tryptophan level. So in the high tryptophan level, what happens? That RNA polymerase is going over, and in high tryptophan level, as the co-translational transcription is happening, the ribosome is chasing the RNA polymerase. So what happens? That in this case, these are the two trip codons. The ribosome doesn't pause in the trip codons because there is abundance of the tryptophan tRNA due to the high tryptophan level. So there is plenty of tRNA, so it just goes through here very quickly. So where it stops is that when it gets into the stop codon of the leader peptide. So this is the leader peptide. This is a stop codon of the leader peptide. So the ribosome actually pauses in here, and see the leader peptide stop codon is in the region 2. So as the region 2 is occupied with the ribosome, the region 2 cannot make a stem loop structure with region 3. And as it pauses in the stop codon, the RNA polymerase moves to the region 3 and 4. As soon as it moves through the region 4, the 3 and 4 forms the stem loop structure, which is the terminator loop, which is followed by the slippery sequence. And as soon as this basically is formed, this is basically the row dependent termination. So the row dependent termination, so row factor, which is the RNA polymerase helicase, we talked about that. That will basically make the RNA polymerase fall off the DNA. So again, this is the terminator loop followed by the poly-U tract. So here, this is the termination of transcription. RNA poly does not transcribe the therapeutic structural gene due to the high tryptophan level. And this is basically what we call as the transcriptional attenuation, depending upon high or low tryptophan level. So after that, there is also another kind of regulation, which are more recent discoveries compared to the black operon and the trip and arabinose operon. And this is a specialized form of transcription attenuation. And this happens due to the folding of the RNA leader sequence, which is basically the riboswitch. And this is similar to the trip, you saw the attenuation. But this basically, the folding pattern alters the response of an effector molecule binding to the RNA. And often the effector molecule is the molecule that is basically produced or basically the end product of the pathway. So for an example, I'm just saying that if tryptophan by itself could do something in here, we will call this as a riboswitch. So let's take a look at these, that how the riboswitches, say for an example, gram-positive bacteria function in transcriptional termination. Like say for example, in Bacillus subtilis, this is actually studied very well. So here what you are looking at, that right here, you have the mRNA start and you have the extensive amount of the secondary structure. And right here, there is a box right here and this RFN box. And this is basically the switch box. And you can see that followed by a stem loop structure, followed by a poly-U tract. So for an example, one of the product of this is a riboflavin. And you know this riboflavin is important because it produces this FMN, which is the flavin mononucleotide. It also produces the FAD, flavin adenine dinucleotide, which is absolutely essential for all these Krebs cycle, electron transport pathway, respiration. So it forms FAD, FADH2, and transports electron. It's very, very essential in many metabolic pathways. But one of the products is also FMN, which is flavin mononucleotide. So what happens that when there is enough amount of FMN or the metabolite is produced, that means there is enough amount of gene expression is going on. The FMN binds to this RFN box. So when it binds to this riboswitch, it changes its structure. And in this condition, you can see that there is a terminator loop basically forms followed by the poly-U tract. So in this case, there is no terminator loop. But in this RFN box, when the gene is formed, that basically alters the secondary structure of the RNA. And due to the secondary structure of the mRNA, this terminator loop forms followed by the U sequence. And this is a classic example of the termination, row-dependent transcription termination. So the RNA polymerase falls off and basically turns off the gene expression. So on the other hand, there is also something which is called a T-box riboswitch. Leader region adopts two configurations for termination or continuous transcription, binding to tRNA to the leader RNA. And basically in this, the charged tRNA indicates the amino acid levels are high and biosynthesis genes are not transcribed. Uncharged tRNA prevents the transcription termination and leads to gene expression. So this is also known as the T-box riboswitch. So these are even finer control that we see in the different types of amino acid biosynthesis pathways. So this chapter is very important because you will know from this chapter that how the genes are expressed and controlled. And the regulation of the gene expression is essential for controlling the different cellular processes. So this is one of the central part of every life form, no matter whether it's a bacteria or eukaryotes, very, very important. And understanding of expression, control, and regulation is one of the central theme of the molecular biology. And know the different control mechanism and know that how each example actually works. And that will pretty much be the chapter 14.